Hello student, myself Ashruti and today we are going to learn about the bacterial cell structure. This topic is under unit 3 for FY Biotechnology student. Okay, so we will start the bacterial cell structure. Before starting the bacterial cell structure, we must know that what bacteria is. So basically, bacteria are the prokaryotic cell. They are differ from the eukaryotic cell as they don't contain the nucleic membrane. Eukaryotic cell possesses endoplasmic reticulum as well as nucleic membrane in which the nucleic material is embedded. But when we are talking about prokaryotes and specifically when we are talking about bacteria, the nucleic material is flowing freely in the cytoplasm. Okay, now this is what about the bacteria and the prokaryotic cell. Today we are going to discuss about the different types of organelles associated with the bacterial cell, and we will start from the most exterior portion of the cell, and that is cell wall. So the cell wall is the outermost layer of the bacteria and it is having direct contact with the external environment. So whatever the bacteria feels about the external sources is because of the cell wall. So cell wall act as a protectant for the bacterial cell. So in this diagram, you can see the red region is nothing but the cell wall is, and how thick that cell wall is. The thickness of cell wall is directly proportional with the rigidity of the cell. Okay, so today we are going to discuss about the different functions of the cell wall, what is the composition of cell wall, and is the cell wall same in both gram positive or gram negative bacteria, or there are certain variations. But before starting the cell wall, actually we will move towards the recap. That is what we were discussed in the last lecture. So we had seen about the microorganism and their different classes. So whenever we are talking about microorganisms, they are divided in two groups, that are prokaryotic cell and eukaryotic cell. The prokaryotic cell means what? The primitive structure, and the primitive structure containing prokaryotic cell is nothing but the bacteria. Okay, so the bacteria is again divided into archaea and eubacteria. We have already discussed that when the earth is evolved and when the life is evolved, okay, initially the archaea bacteria developed. They do not contain the thick layer of peptidoglycan, or they do not contain the similar structure like eubacteria, but the archaea bacteria can be defined as an ancient bacteria. Okay, while the eubacteria are known as recent bacteria which possesses well developed structure. Again, microorganisms are divided in eukaryotes. Okay, and in eukaryotes we can include the examples like algae, fungi as well as protozoa. So today we are going to discuss about prokaryotic bacterial cell, prokaryotic eubacterial cell. Yes. So today's topic will focus on bacterial cell wall, how the cell wall is made, okay, and what are the main functions of the cell wall, why that cell wall is there in bacteria. Then we will discuss about the peptidoglycan. It is a part of a bacterial cell wall. And then lastly we will discuss about the cytoplasmic membrane, okay, which is exactly having the beneath structure of cell wall. Now, whenever we are talking about the structure of bacteria, we can understand the bacterial structure by two different organelles, the exterior organelles as well as the interior organelles, and we will start from the most exterior organelles today. Okay, so the exterior structures of bacterial cell include cell wall, flagella, cytoplasmic membrane, capsules, pili as well as slime layer. And the internal structure or the interior structure of bacteria may include the cytoplasm, endospore, Nucleoids and cell inclusions. Whenever we are talking about cell inclusion, it means that the organelles which are there present in the cytoplasm. That is what comes under the interior structure of the bacterial structure. Okay, and today we are going to focus on exterior structure and the cell wall. So let's start with the cell wall. So before starting the cell wall, let's see how the cell wall is important. So here there are some functions of the cell wall. The first function of the cell wall is that it gives the bacteria a proper shape. We had seen that bacteria are having two main shapes, that is cocci and rod, and those shapes are because of the cell wall. Okay, we can understand the bacterial cell wall relationship with the shape of the bacteria by taking a very simple example. See, water doesn't contain any shape, okay, and cytoplasm possesses almost 80 to 90% of water. So cytoplasm we can categorize in the form of water. So if the water is put it in a glass, okay, it will take the shape of that glass. The same way, okay, cytoplasm take its own shape with the help of cell wall. The next important function of the cell wall is that it protect the cell from expanding or bursting. Okay, there are various reasons because of that cell faces lots of difficulty to survive, and bursting or expansion of cell is one of them. So bacterial cell wall helps the bacteria to protect itself from bursting, and that bursting take place because of the osmolar imbalance. The third important function is that it protects the bacteria from the external environmental changes. The external environmental changes means what? If suppose the temperature is too high or too low, if suppose humidity is too high or too low, if pH is getting variation, okay, so in such cases bacterial cell wall helps the bacteria to survive. Okay, the next important function is what? It also responsible to protect the bacteria from the external antimicrobial drugs. Okay, we all know about the antibiotics, and all antibiotics are having their specific target cells. Means what? Antibiotics are having their mode of action. Now, as per the Darwin's rule, every cell try to survive, and that survival of bacteria is because of the cell wall. Cell wall also plays very important role when bacteria wants to survive. Then comes, it provide the rigid platform to the bacterial cell surface. We have seen the various parameters which we use to disinfect the surfaces, okay, in our practical. We have seen the aseptic condition, we have used the Dettol or disinfectant to clean the platform. So why we are doing this? To avoid the germs. Okay, it means that such type of chemicals are having their adverse effect on the cell. Okay, and cell also defend such type of disinfectants through various responses. Cell wall also help the bacteria to survive against such type of disinfectants as well as the exterior environmental as well as man-made exposure. Okay, it also responsible for the cell division. So when bacteria divides, when bacteria reproduce, at that time also cell wall plays very important role. So this is how the cell wall is very important. Now we will move towards very important point, that is the structure as well as assembly of cell wall. We have talked about the functions of cell wall and how important it is, but we must know that what actually the function of cell wall is, through which material the cell wall is made. So simply, for bacteria, the cell wall is composed of peptidoglycan, which is also known as murine. Murine. Okay. Now, as per the name, peptidoglycan, it contains peptide as well as carbohydrate molecules, because we named it as peptidoglycan. Okay. Now, what is it? It is the mesh-like structure. It is the gauze-like structure. Mesh-like means what? The molecules of the peptidoglycans are arranged in such a way that it can create a mesh on the cell. It can create a mesh on the cell, and through that mesh, the bacteria get protected. Okay, so let's see the structure of the peptidoglycan. So peptidoglycan is made with the N-acetyl glucosamine and N-acetyl muramic acid. They are linked by beta-1,4 linkage. N-acetyl glucosamine and N-acetyl muramic acid linked with each other by beta-1,4 linkage, and through that linkage they create a mesh like structure surrounding the bacteria. Okay, these subunits are nothing but the monomers or molecules of glycans as well as peptide. Okay, the N-acetyl muramic acid is associated with the N-acetyl glucosamine with the peptide bond, and the bond we will discuss over here. Through this diagram, we can understand the structure. So, this is what the structure of N-acetyl glucosamine and N-acetyl muramic acid, they are arranged with each other by peptide bond. Now here the balls which are orange in color are known as N-acetyl glucosamine, okay, denoted as N-acetyl glucosamine, and the green color balls are N-acetyl muramic acid. Now we can see the small blue, orange, purple color balls. They are nothing but the different types of amino acids, and these four amino acids are responsible to make a peptide bond between two chains of N-acetyl glucosamine and muramic acid. Okay, and the amino acids are L-alanine, D-glutamine, L-lysine and D-alanine. Okay, so these amino acids are responsible to create cross-linking between N-acetyl glucosamine and N-acetyl muramic acid, and in such a way, by forming a very rigid structure, they protect the cell. Okay. Now let's focus on the gram-positive cell wall. We had already discussed about the gram-positive and gram-negative bacteria, okay, while we had performed the staining procedure. Now what is gram-positive bacteria and what is gram-negative bacteria? Gram-positive bacteria contain the peptidoglycan, while gram-negative bacteria contain very thin layer of peptidoglycan. So in general, the gram-positive bacteria contain very high amount of peptidoglycan, and because of that, they contain thick layer of peptidoglycan or murine on their cell surface. Almost 90% of the cell wall of gram-positive bacteria contributed by the peptidoglycan. Okay? It means that their linkages are very thick. Their mesh is very thick. Okay. All of this combines together to create incredible strong cell wall in gram-positive bacteria. One more important parameter which is present in gram-positive bacteria is nothing but the teichoic acid. Okay, pronunciate it as teichoic acid. Teichoic acid is anchored from the cytoplasm and comes out the cell wall. Okay, teichoic acid also plays very important role in gram-positive bacteria with respect to their protection, with respect to their cell function. As well as teichoic acid is only and only present in gram-positive bacteria. It is absent in gram-negative bacteria. Teichoic acid is nothing but the glycopolymer which is embedded with the peptidoglycan layer, and it is anchored in the cytoplasmic membrane. It generates the negative charge on the cell surface and also responsible for the proton motive force. It means that when bacteria tries to balance the charge in their cell, at that time teichoic acid is responsible for the exchange of positive and negative ions. Okay, and it continuously responsible to create the net negative charge on their cell surface. It contributes for the rigidity for the gram-positive cell. It participates in cell division. It is also having its important role in cell resistance. Now here we are talking the cell resistance. So whatever the factor which is there to just kill that bacteria, at that time teichoic acid help the bacteria, or teichoic acid help the gram-positive bacteria to resist that factor. That factor can be temperature, that factor can be chemical. Okay. Teichoic acid can either be covalently linked to the peptidoglycan, and it can also called as wall-teichoic acid or WTA, or connected to the cell membrane via a lipid anchor, in which case it is referred to as lipoteichoic acid. It is referred to as lipoteichoic acid. Now why lipo? Because the cytoplasmic membrane is made with nothing but the lipid molecules, and as it anchored in the lipid membrane, that's why it is known as lipoteichoic acid. Okay. So peptidoglycan is relatively porous as it is mesh-like structure. That's why the pores must be there in a mesh. Most substance can pass through the gram-positive cell wall, but some nutrients requires exoenzymes. Okay. Why some nutrients require exoenzyme? Because some nutrients are larger in size, and if the mesh or the pore of the peptidoglycan is smaller in size comparative to the exterior large molecule, at that time that mesh doesn't allow entry of that complex large molecule, and in such cases bacteria needs to synthesize certain enzymes outside their cells, and those enzymes responsible to cleave that large molecule and convert it into smaller form. As soon as the molecule become smaller from larger, the molecule easily pass through the mesh-like structure. Okay, and as the enzymes are synthesizing outside the cells, they are known as exoenzyme. So uh, every content or everything has its own limitation. The same way, peptidoglycan also possesses such type of limitation, that the larger molecule than the pore doesn't allow to pass from it. At that time, bacteria must have to synthesize the enzyme. Through that enzyme, the cleavage of that larger molecule take place, and as soon as it converts into the smaller molecule, it will easily pass through the mesh-like structure or the peptidoglycan. They function outside of the cell, the exoenzymes, and break down the large molecule to convert it into the smaller form, and then the peptidoglycan allows the passing of that smaller molecule. Now here we can directly see the difference between gram positive and gram negative cell wall. Now here the rightmost side is gram positive cell wall and the left one is the gram negative cell wall. We can see how simple the gram positive cell wall is there and how complex gram negative cell wall is there. The reason is that gram positive bacteria possesses very thick layer of peptidoglycan. Okay, so we can see the different layers of the peptidoglycan which are present in gram positive bacteria, right? But gram negative bacteria possess very thin layer of peptidoglycan. This is how actually the gram positive and gram negative bacteria differs from each other. Now, what about their uh, other functions? Means gram positive possesses cell wall and it contains its own functions, like it is responsible to give a shape, it is responsible to give a rigidity, it is responsible for resistance and for survival. Then what about gram negative bacteria? How they survive? Now, in gram negative bacteria, instead of peptidoglycan thick layer, they contain thin layer of peptidoglycan and contain lipopolysaccharide. Okay, they contain lipopolysaccharide. So this is the second difference between gram positive and gram negative bacteria. The first one is about the thickness of peptidoglycan. Okay, so gram negative bacteria contain lipopolysaccharide which are made with the lipopolysaccharide O antigen and lipid A antigen, which makes the gram negative bacteria protective against the environment. Now let's discuss about the cell wall of gram negative bacteria. As we have already seen in the last picture, the gram negative cell wall is more complex than the gram positive bacteria. Instead of peptidoglycan thick layer, it contains very thin layer of peptidoglycan, as well as it contains the lipid bilayer or lipid polysaccharide. Okay. Now that lipopolysaccharide, but obviously as per its name, they are made with the lipid and the glycans, that are polysaccharides. Okay. Now the membrane of that gram negative bacteria is rich in lipid. The outer membrane is impermeable serve as a barrier towards various type of toxins and the unwanted material. The barrier for external chemicals and enzyme uh, act as a lipopolysaccharide. Lipopolysaccharide is a bilayer structure and it is also responsible to function as endotoxin. Now, gram-negative bacteria are developing, or they usually develop different types of resistance towards different environmental condition, and such type of resistance is because of the lipopolysaccharide. The second important thing about gram-negative cell wall is that lipopolysaccharide itself act as an endotoxin. Itself act as an endotoxin, and by producing endotoxin, it protect the cell as well as harm the host. Host means what? Wherever the cell is present in the host. Uh, Now the lipopolysaccharide layer is divided in three groups: the lipid A, then polysaccharide and polysaccharide O antigen. The lipid A firmly embedded in the membrane, polysaccharide located at the membrane of the surface, and polysaccharide O antigen which extend like viscers from the membrane surface into the surrounding medium. Okay, and such assembly is because to protect the cell from the environment. Okay, they are having receptors bacteriophage attachment, means what? They are very susceptible for the bacteriophage. Outer membrane allows smaller molecules like nucleoside, oligosaccharide, monosaccharide, peptides as well as amino acid. Now we will move towards the cytoplasmic membrane. Cytoplasmic membrane is exactly beneath the cell wall. Okay, as we have seen in previous structure, what is the exterior structure of the cell? The outermost structure is cell wall, and the next structure from the cell wall is nothing but the cytoplasmic membrane. And beneath the cytoplasmic membrane, there is presence of cytoplasm. Now, how that cytoplasmic membrane is important? So, first thing is that it is important because it protects the cytoplasm. It disallow the exposure of cytoplasm to the environment. That's why cytoplasmic membrane is very important. It is also known as plasma membrane or bacterial cytoplasmic membrane. Is composed of a phospholipid bilayer and thus has all of the general functions. The function of the cell membrane include acting as a permeability barrier for most molecule and serving as the location of the transport molecule into as well as outside the cell. Whenever we are talking about permeability, it is always associated with the osmolar balance. So, whenever we have discussed about osmotic shock or osmolarity, it must contain the semi-permeable membrane. And through that semi-permeable membrane only, the molecules concentration remain balanced in both the side. Okay. Now let's discuss about the function of the semi-permeable membrane. If suppose we are having one solution which contain very less amount of H2O molecule and very high amount of sugar molecule, okay, and the other side we contain very low amount of sugar molecule and very high amount of H2O molecule. When we mix the solution, but by putting semi-permeable membrane uh, between them, at that time what happened, that semi-permeable membrane, what it will do, it will allow the either water molecule to pass from the semi-permeable membrane and goes to the other side to balance the sugar molecule concentration, or it allows transfer of sugar molecule to the other side to balance the water molecule concentration. So semi-permeable membranes aim is what? To maintain the concentration of molecule present in water. The same way, cytoplasmic membrane is responsible to maintain the balance of molecule. Means what? It doesn't allow the cell to get shrink because of lack of water, or it doesn't allow the cell to burst because of entry of lots of water molecule. Okay. So yes, this is what the structure of cytoplasmic membrane exactly beneath the cell wall. Okay. So the green color line is the green color line is nothing but the cytoplasmic membrane, and the red line is nothing but the cell wall. Okay, and the structure which is squared, from there the semi-permeability of the cytoplasmic membrane take place. The bacterial cytoplasmic membrane is a fluid phospholipid bilayer that encodes the bacterial cytoplasm. Phospholipid structure is nothing but a assembly of molecule which consists of a polar phosphate as well as non-polar phosphate. Okay, now what is the meaning of polar or non-polar? Polar molecules are generally having hydrophilic in nature, means they are having their affinity to bind with the water molecule, while non-polar molecules are the molecules which are having phobia towards the water. Okay, this assembly is because to maintain the proper uh, shape to the cell, okay, and to avoid the dissolving of the cell. Okay, because of that the polar and non-polar combination is there. Now wherever the polar molecule is there, it is located as circle in shape, while non-polar molecules are thread-like in shape. Okay, they are associated with each other and such bilayer is present in cytoplasmic membrane. The unsaturated fatty acid results in kinetics in the hydrophilic tails. The plasma membrane consists of two adjacent layers of phospholipids. The cytoplasmic membrane is semi-permeable and determines which molecule actually the cell need, and which molecule should go inside the cell and which molecule should go outside the cell. So this is what the structure of cytoplasmic membrane or plasma membrane, a very well known structure which is also known as fluid mosaic model, which we have already seen in our uh, past years. Okay. Now what is the concept of such type of assembly is? We can see the circle, red circle as well as red threads. The red circles are nothing but the polar regions of the phospholipids. Okay, they are having affinity towards the water, and because of that they are arranged like this. Okay, now why they don't get dissolved very easily in water? Because they are having affinity, because they are immediately linked with the non-polar hydrophobic phospholipids. Okay, non-polar hydrophobic phospholipids which doesn't allow the polar molecule to get dissolved in water. Okay, and because of that they create a proper bilayer shape. Okay, apart from the lipid bilayer, the cytoplasmic membrane also possesses various type of arrangement. It include glycolipids, it include oligosaccharide, it include peripheral protein which are responsible for different functions. It also possesses hapenoids, and hapenoids help the bacteria or help the cell to maintain a proper permeability. Okay, so hapenoid molecules also act as a signals, okay, which immediately inform the cell which now cell needs what. Either it need water or either it need high concentration of molecule. So this is how the assembly of plasma membrane is there in bacterial cell. It contain hydrophilic ends of the membrane, it contain floating in a lipid bilayer, it contain hydrophilic fatty acid chains. Okay. So all molecules and atom possesses kinetic energy, and cytoplasmic membrane also possesses the kinetic energy, and it also responsible to maintain the concentration on both the side, means inside the cell, inside the bacterial cell, that is in cytoplasm, as well as outside the cell, that is in environment. Okay, as soon as we put the cell in very highly concentrated solution, what is the expectation is the cell become shrink. But cytoplasmic membrane tries a lot to protect the cell from the shrinkage by allowing the by disallowing the water from inside the cell to outside. But then though, because of lack of water, the cell get shrinked. Okay, and that is what the reason we generally add sugar to preserve our fermented food. Okay, if we are making any type of sweet or any type of long term food, okay, What we add in it? We either add sugar, we either add salt into that. Okay, what that sugar and salt will do generally in pickles? What they do? They protect the fermented food. How? By disallowing the microorganism. At that time, the because of the semi-permeable membrane of that semi-permeable nature of that cytoplasmic membrane, what happens? Because of high salt concentration, the cell started to shrink. So this is how the cytoplasmic membrane function. The net movement of these particles will therefore be down their concentration gradient from the area of higher concentration to the area of lower concentration. Okay, even the diffusion is also responsible because of the cytoplasmic membrane. Now what is diffusion? If suppose we put a ink pen on a paper, on a blank paper, what happened? The ink started to spread. Why? Because that blank paper is nothing but a porous material, and through that porous material the ink is migrating from one pore to another, then another, and then another. Okay, so this procedure is nothing but known as diffusion. Okay, this procedure is known as diffusion. So for diffusion, we need a support, like to diffuse. If suppose ink wants to diffuse, it need a paper. The same way, if certain material want to diffuse inside the cell, or if the cell needs any diffused material, at that time it must contain a matter. Okay, it must contain a matrix, and cytoplasmic membrane provides the matrix or the support for diffusion of various materials. So yes, we have discussed lots of things, and I am also expecting from you to answer some questions like what are the functions of the cell wall, what is the difference between gram positive and gram negative cell wall, and what is the role of cytoplasmic membrane whenever cell wants to function. Okay, so such three questions I am expecting from you to answer in this next lecture. So yes, let's do the revision of today's lecture. Today we have seen the peptidoglycan as well as the N-acetylglucosamine and N-acetylmuramic acid assembly, which combinedly make a cell wall. Next, we have seen the differences between gram positive and gram negative cell wall, how they differ from each other, how the peptidoglycan layer is having its own thickness, different thickness in gram positive and gram negative cell wall, as well as we have seen the various function of cytoplasmic membrane, and two important function of cytoplasmic membrane is nothing but the permeability of the cell as well as transport of molecules. Okay, so this is what we have learned in today's lecture. I am expecting from you to understand every concept from this lecture. In next lecture, we are going to discuss the other parts of the bacterial cell. We will discuss the flagella as well as endospore. We will discuss the capsule as well as we will discuss the inclusion bodies which are there present in the bacterial cell. They are having their own function. It is not necessary every bacteria contain capsule, every bacteria contain flagella, and every bacteria contain endospore. Okay. So depending upon such type of organelles also, bacteria are classified in various categories. That is what interestingly we will discuss in our next lecture. Thank you.